Hello and welcome to Chapter 9. We're going to talk about genetics in Chapter 9. And your Media Under the Microscope article at the beginning of this chapter is talking about the Defense Advanced Research Projects Agency, or DARPA, and their research into manipulating human genes to make individuals more resistant to things like disease or to injury, etc. And so we're really talking about creating the perfect super soldier in the DARPA's research. So something like Captain America. Could we insert or manipulate genes that would actually allow this to occur? And that's got a lot of questions to it, both from a technical standpoint as well as ethical standpoints. So let's look at genetics. Well, what is genetics? It is the study of the inheritance or heredity that explores the transmission of the biological properties or traits from parents to offspring. It also looks at the expression and variation of these traits and the structure and function of the genetic material in which this genetic inheritance is encoded. And how does this material change over time and generations? So, what makes up an organism? Well, certainly all of the cells, but what makes up and directs the function of the cell will be the genetics within the chromosomes or the DNA. Genome is actually defined as the sum total of all genetic material within an organism. Most exist in the form of chromosomes. Some may appear in non-chromosomal sites, however, such as plasmids in some of the bacteria and fungi, and organelles such as within the mitochondria and chloroplasts. Genomics is an all-encompassing umbrella term that literally studies the organism's entire genome. This would include both the DNA and RNA structure and function. So, just a reminder, where is the primary DNA found in eukaryotic cells? Well, the inheritable form is within the nucleus in the form of those chromosomes. But remember, the chloroplasts and the mitochondria also have their own circular DNA, even though they're not shown as circles here in the picture. And in some eukaryotic cells, such as in some fungi and protozoa, they actually also contain plasmids. In the prokaryotic cells, they typically have that single circular chromosome, and they may or may not have plasmids. And then in the viruses, again, it's either a DNA carrying virus or an RNA carrying virus. The chromosome is a discrete cellular structure composed of a neatly packaged DNA molecule. Eukaryotic chromosomes are located primarily in the nucleus. They vary in number from a few to 100. They can occur in pairs or being diploid in number or singles as in haploid. And they are linear in appearance. Whereas the bacterial chromosome usually is a single circular and haploid chromosome. There are a few exceptions to this, however. So a few have some multiple circular chromosomes, and even a fewer actually have linear chromosomes. Genes are the basic informational packets. They are what encode for a product of the genes. Classic inheritance or classic genetics actually refers to genes as a functional unit of heredity. Molecular and biochemical genetics define genes as the site on the chromosome that provides information for a certain cell function. And so we try to combine these with the current preferred definition of genes. It's a segment of DNA that contains the necessary code to make a protein or an RNA structure. Now, genotype are the actual genes that one possesses. It is the sum of all types of genes constituting an organism's distinctive genetic makeup. It's the actual genes that one possesses. whereas the phenotype is the physical expression of the genotype that creates certain structures or functions within the organism. Now, genomes vary in size. For example, E. coli has that single circular haploid chromosome that has 4,288 genes. If we unwind that single circular chromosome, it's about 1 millimeter long. It's 1,000 times longer than the cell itself, with E. coli being at about 1 micron in diameter. So we have to wad it up in order to get it into the cytoplasm. In the human genome, there's approximately 23 to 25,000 genes found on the 46 chromosomes that we possess. So we can actually look at this chart and see that there are differences within the number of chromosomes, and there'll be a difference in the number of genes that each organism possesses. So for example, corn has 20 chromosomes, while soybeans actually have 40 chromosomes. We're at 46 with about 3.2 billion base pairs, and we'll do a little bit more comparison here in just a moment. So let's look at this. So for example, the T2 bacteriophage has 170,000 base pairs within its genome. It is a DNA carrying virus, and so it's actually a fairly small genome overall. If we look at E. coli, on its singular circular chromosome, there are 4.6 million base pairs found in those 4,000 some genes. In Drosophila, or the common fruit fly, they have 8 chromosomes, and in those 8 chromosomes, they have 130 million base pairs. In humans, in the 46 chromosomes we possess, we have 3.2 billion base pairs, but we actually are not the top of the food chain in this case. There is actually a plant known as the canopy plant that has 200 some odd chromosomes and 150 billion base pairs in those 200 chromosomes. Now, the basic unit of DNA is what's known as the nucleotide, or sometimes you'll see it printed as nucleoside. Each nucleotide consists of a phosphate molecule, deoxyribose sugar, which is a 5-carbon sugar, and one of 4 nitrogenous bases. And the 4 nitrogenous bases of DNA are going to be adenine, which always pairs with thymine, and guanine, which always pairs with cytosine. DNA is a double polynucleotide helix. So here's a nucleotide, the phosphate, the deoxyribose sugar, and a nitrogenous base. Each nucleotide then will be linked to another one and another one, forming a chain, and then we have an opposite side where the bases face each other and form basically the rungs of the ladder. The sugar phosphate backbone actually creates then the side rails of the ladder. This double helix is in what we call an anti-parallel arrangement. One side of the helix runs in the opposite direction of the other. And so what we'll find is that on one side, if we start at the top of the molecule, we go from the 5' to 3' in one direction, and the opposite strand at the top begins at a 3' and runs to a 5'. 5' actually means that the sugar phosphate backbone starts with a phosphate group, whereas the 3' is the sugar end. This is a significant factor in DNA synthesis and protein production because when we copy DNA and RNA, the new copy will always be synthesized in the 5' to 3' direction only. And so we can actually see then that when we copy the double strand here, we actually are going to make the copies in different directions because we can only, again, make the new strand in the 5' to 3' direction. And so this makes each one of these strands different from each other, and we'll talk about that a little later. The overall replication process of DNA requires some enzymes, and so we need to talk about these first. Helicase actually unzips the DNA helix, separating the two halves and forming what's known as the replication fork, where the two strands are separated. RNA primase will actually synthesize an RNA primer. DNA polymerase III adds new DNA bases to the new DNA chain, proofreading for the chain for mistakes. And DNA polymerase I actually will remove the primer, close the gaps, and repair any mismatches. Now, I'm not going to really make a big difference between polymerase III and I, but just simply to say DNA polymerase makes DNA. DNA ligase will be the final binding of NICs in the DNA sugar phosphate backbone during both synthesis and repair. And I want to come down here to DNA gyrase, also known as topoisomerase II. DNA gyrase will actually unwind or remove the supercoiling ahead of the replication fork so that helicase then can separate the two strands and they will be parallel to each other. The overall replication process is what we call semi-conservative in nature. This means that each daughter molecule of DNA is identical to the parent in composition. Neither, however, is completely new because each of the copied DNA will have one parental strand, which was the template strand of the original DNA, and one new strand. Now, DNA polymerase III synthesizes a new daughter strand of DNA using the parental strand as the template and simply matches A's and T's, C's and G's as it reads along it. The DNA molecule must first be unwound and separated. The unwinding will occur with DNA gyrase, and separation will occur with helicase. DNA polymerase can only add nucleotides to an existing chain and cannot begin synthesizing a chain of nucleotides. It can only add nucleotides in a 5' to 3' direction. And it's one of these few times that we can say always, always, always. The new strand of DNA or RNA will always be made in a 5' to 3' direction. The replication fork formed by helicase is where the two strands will be unwound and separated, and this is where the replication will be taking place. Each circular DNA molecule in prokaryotes will then therefore have two replication forks moving in either direction from what's known as the origin. The RNA primer is a small length of RNA that is inserted initially during replication. The reason for the RNA primer is because DNA polymerase can only start adding new bases to an existing length of nucleic acid. And so the RNA primer has to be laid down first in order for the DNA polymerase to actually attach and begin laying down DNA nucleotides. Now, because the anti-parallel arrangement will mean that the two strands are made in opposite direction, we can call one strand the leading strand. This strand of new DNA will be synthesized continuously in a 5' to 3' direction towards the replication fork. The lagging strand, because it is running in the opposite direction, however, cannot be made towards the replication fork. It is made actually away from the replication fork, and it is done so in short segments that then will be tied together by DNA ligase. The Okazaki fragments are these short segments, and by short, anywhere from 1,000 to 5,000 nucleotides in length, and these short segments will be synthesized away from the replication fork in the 5' to 3' direction, and eventually the primers will be removed and ligase will seal up all of the nicks. So let's take a look. Here's our replication fork caused by helicase. The unwinding is being unwound with DNA gyrase. So let's look at each one of these. The original template strand on this side, actually if we looked at this end down here, is a 3' end. So what this means is the opposite strand that will be made will be running from a 5' to 3' direction towards the replication fork. And so therefore, this is the leading strand. But the opposite parental strand was actually seen from the 3' to 5'. So this end out here is a 5'. So we can't start here and go this way because this would be a 3' end. So what the RNA primase will do is we'll come down here on the length of the lagging strand, lay down the RNA primer, and then we make that short Okazaki fragment from the 5' to 3' end. But then we run into the previous RNA primer, and that will kick then our DNA polymerase off. Eventually, another DNA polymerase 1, will come in and remove the RNA primers, and then DNA polymerase then will fill in all of the extra DNA nucleotides, and ligase then will seal up the backbone. So if we're looking at a circular chromosome, we actually have an origin up here originally, and we'll have a replication fork running this way with the leading and lagging strand, and a replication fork running this direction. Eventually they meet and we actually see a termination, and now we have the two daughter strands where we have one parental and one newly synthesized strand in each one. Replication of the eukaryotic DNA is similar to replication of prokaryotic DNA. There are actually some more DNA polymerases that we're not going to discuss, and replication proceeds in both directions, but now from actually multiple origins along the long axis of the chromosome. Topoisomerases are used to relieve the tension in the strand from these origin points and to recompact the molecule when it's fully replicated. So, what is a gene? A fundamental unit of heredity, a site on a chromosome, a segment of DNA that codes for a protein, or all of the above. Match the base pair with its appropriate nitrogenous base. So A pairs with what? T pairs with, G pairs with, C pairs with. And define the term semi-conservative replication. Now, the central dogma of genetics is this, and it's a very simple concept. The DNA will be transcribed or copied to RNA, the messenger RNA. The messenger RNA, along with transfer RNA and the ribosome, will then be used to produce proteins. And this is known as translation. Think of it this way. What if your text was written in Italian? Most of us don't read Italian, but can we make a copy of it? Well, we actually can, and that is simply transcription. Scribe means to write, so we make a copy of it. Does it mean anything, though? It doesn't mean anything until we have a translator. And really, in analogous respect, the ribosome is the translator, translating messenger RNA using tRNA to produce proteins within the cells. But the central dogma is really incomplete. First of all, there are a wide variety of RNAs that are used to regulate gene function, and we'll mention a few of these as we go through here in a few moments. There are many genetic malfunctions that could cause human disease and are found in these regulatory RNAs, but not in the genes for the proteins themselves. And the DNA that codes for these crucial RNA molecules were once called junk DNA, or DNA that didn't code for any amino acid within a protein. We now understand, however, that these may actually be very important in regulating gene expression within eukaryotic cells. So, from the DNA, we are going to transcribe that to a messenger RNA. We then are going to use transfer RNA and ribosomal RNA in order to translate the message within the messenger RNA into a series of amino acids. The sequence of amino acids will determine how this protein folds up, and how the protein folds up determines its function. Now, what is the connection then between the code in the DNA and an organism's traits? Well, the protein's primary structure determines, again, how the protein is going to fold up. How it folds up determines what its function is. And the proteins are ultimately going to determine the phenotype within the cell, and the cells then are going to make up the phenotype that we see in the larger multicellular organisms. We now actually have a fairly new area of study known as proteomics, and this is actually the study of an organism's complete set of expressed proteins from the genetic code. So DNA really is a blueprint that tells the cell what kind of proteins to make and how to make them. So we'll see with the double-stranded DNA, and what we'll see is within the DNA we will have 3 nitrogenous bases in sequence, and these are known as the DNA triplets. Each one of these will code for a nucleotide within the messenger RNA, and the 3 nitrogenous base sequence within the messenger RNA is known as a codon. Each codon will code for a single amino acid. So participants in transcription and translation. Messenger RNA will be transcribed from the DNA. Transfer RNA's job is to bring in amino acids to the ribosomal complex. Regulatory RNAs, including small interfering RNAs or microRNAs, are going to regulate how often the gene is being copied and how often it is going to be closed where it is not being copied. And then the ribosomal RNA that actually makes up the ribosomes themselves. There'll be several types of enzymes involved. One, certainly proteases in order to make the peptide bonds between the amino acids. And of course, all of the amino acids themselves. So this is just the table of the RNA. So again, messenger RNA carries the message from the DNA master code to the ribosome. Transfer RNA is going to bring the amino acids to the ribosome during translation. Ribosomal RNA makes up the ribosomes itself. The microRNA, antisense riboswitch, and the small interfering RNAs are regulatory RNAs, and they actually regulate the gene expression and coiling of the chromatin. And then we're going to talk a little bit later on about the ribozymes and spliceosomes found in eukaryotic cells that remove parts of the messenger RNA, what's known as the introns, before translation actually occurs. So here's my messenger RNA that we copied. Messenger RNA is a single strand of nucleic acid, and we have adenine, guanine, cytosine, but now what we have is uracil in place of the thymine. The transfer RNA forms kind of this weird clover look leafing thing shape. It's kind of weird. And we have two important areas. One is right up here where the 5' and 3' end fold on itself, and this is the amino acid attachment site. This is where whatever amino acid will attach, and what amino acid attaches here is determined by the opposite end, which is known as a 3-nitrogenous base sequence known as an anticodon. So for example, if our first codon of messenger RNA is AUG, I simply make the complementary bases. So A goes to U goes to A, G goes to C, and that's the anticodon of the messenger RNA codon. Now I will tell you that AUG codes for the amino acid methionine. So if a transfer RNA has an anticodon of UAC, it will attach to a methionine molecule out here in the cytoplasm. So RNA is a single-stranded molecule. It does exist in a helical form. It can assume, however, secondary and tertiary levels of complexity. And so just like we see in the transfer RNA. It contains uracil as a nitrogenous base in place of thiamine that pairs with adenine. And the sugar in the sugar phosphate backbone is ribose sugar instead of deoxyribose. Messenger RNA is the transcript of the structural gene or genes that was seen in the DNA. Messenger RNA will be synthesized in a process similar to the synthesis of the leading strand during DNA replication. The new strand of messenger RNA will be made in a 5' to 3' direction by an RNA polymerase. The codons are the series of 3 nitrogenous bases that hold the message of the transcribed messenger RNA. Transfer RNA contains the sequence of bases that form hydrogen bonds with complementary sections of the same tRNA strand, and that's what allowed it to fold up in that clover leaf-like shape. The anticodon found at the bottom loop of the clover leaf will determine what amino acid will be picked up by that transfer RNA and attached to the amino acid site. The ribosome will form with the two large and small subunits coming together to form the complete ribosome. And remember, 70S ribosomes are found in prokaryotic cells, while the 80S ribosomes are the primary ribosome in eukaryotic cells, with mitochondria and chloroplasts, though, also having 70S ribosomes. The ribosomal RNA will form complex three-dimensional figures that contribute to the structure and the function of the ribosome, which is to make a protein. It's the interaction of the ribosomal RNA and protein to create the two subunits of the ribosomes that come together. Once the two subunits come together, they actually will form 3 sites within the ribosomal complex. The A site is going to be where the amino acids come in. This will be where the transfer RNAs move in, matching the anticodon of the transfer RNA to the codon of the messenger RNA. The P site is where the peptide bond will be formed. So whatever amino acid this is will then form a peptide bond with the amino acid in the A site. And the ribosome will then move down the messenger RNA. Once the amino acid in the P site is transferred to the amino acid in the A site and the ribosome moves down, this now empty transfer RNA will enter the E site, which is the exit, and will eventually exit out to go back to pick up another amino acid out in the cytoplasm. So, transcription initiation begins on the DNA at the beginning of the gene, and there are several factors that are involved in actually allowing the gene to be opened. We're only going to transcribe one side of the DNA, And so what we'll find is the template strand beginning at a 3' end, because how are we going to make the messenger RNA? We're going to make the messenger RNA in a 5' to 3' direction. There will be a promoter site that basically allows RNA polymerase to bind to that template strand. Then we simply match RNA bases to the DNA bases from the 5' to the 3' direction, and this is known as elongation. And eventually we'll reach a termination segment, which just simply tells the RNA polymerase that's all of the gene. And we actually will kick off the RNA polymerase, the DNA will bind back together, and now we will have this RNA strand that has been made. So the messenger RNA nucleotides then that are transcribed from DNA will then be read in codons, or again these groups of 3 nitrogenous bases. The codon indicates which amino acid will be added to the growing polypeptide chain in the ribosomal complex. Except for a very few cases, this code is universal for bacteria, archaea, eukaryotes, and even the viruses. So if I have UC, does it matter what the third letter of the codon is? No, it's going to code for serine. So if I've got UC, that's it. We've got serine, and that's the only thing we can have. Doesn't matter what the third letter is. So this is known as the wobble. And this redundancy and wobble within the code allow for errors to occur in the genetic code without causing changes in the protein. So here I have my DNA, and I'm going to tell you that this end starts at a 3' end, this one starts at a 5' end. So this one will be my template strand. When I make my copy using RNA polymerase to make an RNA, T goes to A goes to U, C goes to G, G, C, A, U, C, G, etc. Then, once I start reading at the ribosome, AUG, the anticodon of the messenger RNA codon is UAC. This transfer RNA then will have picked up methionine out in the cytoplasm, and it will bring it into the ribosome and actually sit in the P site to begin with. And so what we're looking for on our messenger RNA strand is the first AUG start codon, and that's where we're going to start translation. If we go to the next codon, CUG, the anticodon GAC transfer RNA will pick up leucine. And if you read your codon chart, CUG codes for leucine. This transfer RNA will sit in the A site. Now, methionine can be transferred to leucine by making a peptide bond. We go down and read the third codon, ACU, that codes for threonine. The anticodon UGA means this transfer RNA picked up threonine. It then now moves into the A site. This moves to the P site. This now empty transfer RNA moves to the E site and will exit the ribosome. And then we just keep doing that. Now you'll notice though, ACU coding for threonine. The next one, ACG, also codes for threonine. Different codon, but the same amino acid in that place. So all the elements that are needed for synthesis of a protein are brought together. So again, let's review. Messenger RNA, the transfer RNA, the amino acids, the ribosome made up of ribosomal RNA. So there's really 3 stages of translation. Initiation, where we find that first start codon of AUG on the messenger RNA. Elongation, simply reading the codons, matching anticodons of transfer RNA, bringing in the amino acids, and forming peptide bonds between each of the amino acids as they come in. and finally termination when we reach a stop codon. So, entrance of the transfer RNAs 1 and 2. And again, the transfer RNA with UAC will carry methionine, and the AUG is in the P site to begin with. That then allows the second transfer RNA to come in and bind to its codon and anticodon together, and it will bring in the second amino acid. A peptide bond will be made between methionine and leucine. And then the ribosome is going to move down one codon. And this now empty first transfer RNA will exit the E site, and we move down. And now we have a third transfer RNA coming into the A site, matching anticodon to codon, bringing in proline. We now make a peptide bond between the leucine and the proline, and then we just keep doing this over and over and over again until we reach a stop codon. The start codon, again, AUG, codes for methionine. So when the ribosome and the messenger RNA come together, the P site is looking for that first AUG codon in the messenger RNA strand. The stop codons, or nonsense codons, when we reach the UAA, UAG, or UGA, there is no corresponding transfer RNA. And in fact, a small releasing factor will then come in and sit at the A site, and that will cause in the ribosomal complex to come apart, the subunits separating, the messenger RNA separating from the ribosome, and the last transfer RNA will separate from the final amino acid that's in the growing polypeptide sequence. Now, translocation is the process of shifting the ribosome down the messenger RNA strand as it reads each subsequent codon. Now, once we have our protein made, we can then do post-translational modifications. Proteins begin to fold up on themselves, determined by the amino acid sequence. The formalin methionine, or just methionine, may be clipped off at the beginning, and other things may be added to the protein. Say, for example, if this is an enzyme, cofactors may be added at that point. Some join with other proteins to form the quaternary structures of the protein. So once we start we find the AUG and we start protein synthesis, the amino acid sequence then is going to determine how this protein is going to fold up, and how it folds up will determine what its function is. And I love this picture. The middle line right here is DNA. And these larger blobs are actually RNA polymerases that are transcribing a messenger RNA strand. And the messenger RNA strand is actually the thin little line in between these little bumps right here. And these little bumps are actually ribosomes then producing the protein. Now, this is actually being seen in a prokaryotic cell because both transcription and translation can occur at the same time in prokaryotic cells because they both occur in the cytoplasm. That is not true in the eukaryotic cell. In the eukaryotic cell, transcription occurs in the nucleus, translation occurs in the cytoplasm. So co-translational translation cannot occur in the eukaryotic cell. AUG, the start codon, codes for a different form of methionine in eukaryotic cells than prokaryotic cells, but I'm not going to really distinguish between the difference. And eukaryotic messenger RNAs only code for one protein. So each mRNA, one protein. Whereas in bacteria, because of something known as operons, bacterial messenger RNAs often contain information for several genes located within a series on one messenger RNA. Most eukaryotic genes do not exist in an uninterrupted series of codons coding for a protein. They have some interspersed DNA that is a non-coding portion of the code, and these are actually known as introns. Here's the way you can remember this. Introns will stay in the nucleus after the messenger RNA is processed. These introns are non-coding portions. So what these introns actually become are some of the regulatory RNAs that we now are just really learning a lot about. Exons are the coding portion of the code that will code for the amino acid sequence. And exons exit the nucleus after the messenger RNA has been processed. The spliceosome or ribozyme is going to be the enzyme that actually recognizes the exon-intron junctions and enzymatically cuts through them. And actually, three things are going to happen to the eukaryotic messenger RNA once it has been transcribed from the DNA. First, on the front end of the messenger RNA, we put a modified guanine nucleotide. This is known as the 5' guanine cap. On the opposite end, the 3' end, we put somewhere between 100 to 250 adenine nucleotides. This is known as the poly A tail. The 5' guanine cap is thought to help the ribosome in the eukaryotic cell find the appropriate end of the messenger RNA to start looking for the AUG codon. The poly A tail, even though it's not involved in the stop codon, may actually just simply stabilize the messenger RNA strand. And then the spliceosome or ribozyme is going to cut out the introns and splice together the exons before the messenger RNA leaves the nucleus for translation. These spliceozymes or ribozymes will loop the introns into lariat-shaped pieces, excise them, and then join the exon ends together to complete the now mature messenger RNA strand that can go through the nuclear pores of the nuclear membrane, go out into the cytoplasm, and attach to the ribosome for translation. And this simply shows, here is our primary messenger RNA strand. Here's our exons and introns mixed together. The spliceozymes or ribosomes will simply loop these things on themselves, and the introns will be released into the nucleoplasms. These, again, introns are going to become these regulatory RNAs that we'll talk a little bit about later on. The exons then are spliced together, the 5' guanine cap will be put on, and the poly A tail will be put on the 3' end, and now this is a mature piece of eukaryotic messenger RNA that can go out for translation in the cytoplasm. Now, viruses consisting of one or more pieces of DNA or RNA will simply inject their genetic code into the host cell. And the host cell then will actually take over reading the viral genome. The viruses, because they are adding genetic material to their host cells, then therefore have the potential for genetically altering the cells in which they infect. And this may be one of the major reasons why certain viruses are linked to the formation of cancer cells. Viral codes tend to be very small, compact, and economical. And there is a diversity of the viral genomes. Sometimes the DNA or RNA can be linear, sometimes it's circular, single molecule in some viruses, segmented in others. And again, they can be double-stranded or single-stranded DNA or RNAs, or they can be retroviruses or other type of viruses that carry their own enzymes that affect the mechanism of the host cell. So, describe the function of each of the following: messenger RNA, transfer RNA, ribosomal RNA, And what enzyme directs transcription? And true or false, more than one codon can exist for an amino acid. And compare and contrast bacterial and eukaryotic transcription and translation, especially the modification of the messenger RNA. Now, in prokaryotic cells, they can contain something known as an operon. And an operon is a coordinated set of genes regulated as a single unit. They have a control region made up of a promoter and an operator, and they always will have a regulatory gene associated with them that may be closely located to the operon or somewhere else in the DNA. These can be inducible or repressible, and the categories are determined by how transcription is affected by the environment surrounding the cell. Now, let me say this. The regulatory gene of these operons is what we call a conducive gene. This means this gene is always on. It's always being transcribed and translating and is producing a regulatory protein for the operon that it's associated with. The operon, though, being inducible or repressible, can be turned off or on basically by the presence of the regulatory gene on the operator within the control site. Inducible operons typically produce catabolic enzymes. The operon is turned on or induced by the presence of the substrate or substrates for which the structural genes encode for enzymes to break down those substrates. So the enzymes needed to metabolize a nutrient are only present or are only made when that nutrient is present in the environment. So the nutrient is going to be the inducer of the inducible operon. Repressible operons usually contain genes coding for anabolic enzymes, and several genes in a series will then be turned off or repressed by the product that's synthesized by the enzymes. So let's look at a couple of examples of these operons, and these are only a couple of examples. But one of the best studied is the lac operon in E. coli. The lac operon has a regulatory gene that codes for a repressor protein, and this protein is capable of repressing or turning off the operon so that the structural genes of the operon are not being transcribed or translated. So the normal position for this inducible operon is off. The control locus, made up of the promoter that recognizes RNA polymerase that would cause transcription to occur, and the operator where the regulatory protein is going to bind and turn off the operon. The structural locus is made up of actually 3 genes in the lac operon, each coding for a different enzyme needed to break down or catabolize the sugar lactose. The repressor protein is allosteric, meaning the binding site for the operator sequence on the DNA and lactose. In the absence of lactose, the repressor protein binds to the operator, thereby blocking transcription of the structural genes by RNA polymerase. The regulatory gene lies upstream of the operator and is transcribed constitutively, meaning that it's always being transcribed and translated in the same amount. So here's the picture. So here's the regulatory gene. It produces at a steady state rate the repressor protein. The repressor binds to the operator, and RNA polymerase tries to bind to the promoter, but basically then runs up against that repressor protein and cannot continue on down and transcribe the structural genes. So the operon is normally in the off mode and does not initiate transcription when the appropriate substrate, lactose, is absent within the cell. But what if lactose is actually in the cell? Well, what lactose will do is it will bind to that regulatory protein on the operator and change its shape, thereby kicking it off of the operator. And what this will do then is allow RNA polymerase to then move down and transcribe 3 structural genes. When lactose has been broken down, then the lactose on the repressor protein will be kicked off, and the repressor can go back to bind to the operator. The other thing is that the repressor protein is not always bound to the operator. It will oftentimes simply release on its own, but then another one of these regulatory proteins would slip on and again stop transcription. It's only when lactose is present then that it will change the shape of the regulatory protein, making it inactive so that we can then read the 3 genes that code for the enzymes that break lactose down. And this is one of these regulatory genes, basically, in the fact that we only turn it on when we need it. It takes energy to make these proteins, and so if we don't have lactose present, why spend the energy in making all these enzymes when they're not needed? Now, a repressible operon is the repressible operons are usually in the on mode. They are being transcribed and translated, but they will be repressed or turned off when the nutrient they're making is no longer required. So excess product of the genes product will actually serve as a co-repressor to block the action of the operon. And so another well-known operon is the trip operon, standing for tryptophan. And the tryptophan actually has 5 structural genes within the operon, and each of these are necessary for making tryptophan. So the regulatory gene, again, will constantly be transcribed and translated and make a repressor protein. However, it's made in the inactive shape, and so it does not bind to the operator, and RNA polymerase can bind to the promoter and transcribe the 5 structural genes, and these enzymes will then make tryptophan. But when tryptophan is in required amounts, it binds to that inactive protein, making it an active repressor protein, and it slips over the operator, thereby blocking RNA polymerase from reading the genes and shuts down transcription of the structural genes of the operon. So it is repressed by the presence of tryptophan. Now, the result of bacteria turning on or off a complement of genes that leads to phenotypic changes is known as phase variation. These genes are heritable and passed down to subsequent generations, and it involves turning on genes mediated by regulatory proteins as described within the operon. Certain antibiotics can affect transcription and translation, and we're going to talk about these a little bit later on in another chapter, but I just wanted to mention a few here. Drugs that inhibit protein synthesis can include the rifamycins, actinomycin D. Drugs that interfere with the actual structure of the ribosome, thereby inhibiting protein synthesis, include erythromycin, spectinomycin, an old drug called chloramphenicol, and lincomycin. So again, we'll come back and talk about these in a little more depth once we reach the antibiotic chapter. Oh yeah, and tetracycline. And the aminoglycosides, sorry. So, describe the differences between an inducible and repressible operon. What are three important features of the lac operon? What triggers induction of the lac operon? And with that, what triggers repression of the trip operon? And true or false, antibiotics that interrupt bacterial protein synthesis could also affect eukaryotic cell protein synthesis. Now, DNA recombination. This is an event in which one bacteria donates DNA to another bacterium to make a new recombined organism or recombinant. The end result is a strain different from both the donor and recipient strain. And plasmids play a big role here in the fact that they are very adept at moving between cells through the pilus. A recombinant then is any organism that contains and expresses genes that originated in another organism. Horizontal gene transfer is the transfer of DNA that results in organisms acquiring new genes that did not directly come from the parent organisms. If the parent passes on the gene to an offspring, that's known as vertical gene transfer. Plasmids, as I said, are very adept at actually moving between cells. They can replicate independently of the bacterial chromosome and may not be passed on from one generation to the next. So they are very easy at allowing transfer of DNA between cells. They're found in most bacteria and even in some of the fungi and a few protozoa. They contain at most a few dozen genes. These genes are not necessary for the survival of the organism, but often carry useful traits. And there's going to be three natural ways in which horizontal gene transfer can occur. Conjugation, which is the direct form of gene transfer, transformation, and transduction, which both are indirect gene transfer. So let's look at conjugation first, and sometimes this is referred to as bacterial sex. Again, bacteria only reproduce asexually, but conjugation is a mode of genetic exchange in which a plasmid or other genetic material is transferred from a donor cell to a recipient cell via a direct connection, and that is the pilus. This can occur in both gram-positive and gram-negative cells. So here we have the F factor transfer, or what's known as the fertility gene. The plasmid of this F plus cell contains the fertility gene that allows it to make a pilus. The pilus then will form the mating bridge. This F minus recipient cell does not have the fertility factor because it doesn't have the F plasmid. The F plasmid will simply be copied in the donor cell and go across the pilus into the recipient cell, and the recipient cell then goes from F minus to an F plus cell. The mating bridge then will be separated, and these two cells will then go on their way. Now this once F minus cell can now grow its own pilus and do the same thing with another cell. High-frequency transfer is another form of conjugation that's a little bit different. Sometimes these plasmids actually are incorporated into the bacterial chromosome. And so we have the integration of the yellow portion right here, which is the fertility gene. And so it will grow a pilus. But what will happen is that we copy only a part of the fertility factor along with the rest of the plasmid and a little chunk of the donor cell's own chromosome. That will be transferred through the pilus to the donor or to the recipient cell. Because we did not get all of the fertility factor, this recipient cell remains an F minus cell. So it can't form a pilus. But what it now has is some of the DNA from the original donor cell that was not present before. So lots of different genes can go along with the fertility factor, and one of the big things that we're very concerned about are resistant genes. And these bear genes for resisting antibiotics or other drugs. And these resisted plasmids are shared commonly among bacteria through conjugation. And so this is one of the things that leads to antimicrobial resistance. The R factors can also carry genetic codes for resistance to heavy metals or for even synthesizing virulence factors, which makes the recipient cell now more virulent or more pathogenic than it was before. Now, transformation. This is actually capturing DNA from the environment. And this is what I refer to as the capturing of a piece of naked DNA. The acceptance by a bacterial cell of small fragments of soluble DNA from the surrounding environment is what transformation is. Well, where did these soluble pieces of DNA come from? They usually come from other cells that have been destroyed. And the cell that is being transformed is under some sort of stress and in threat of actually not being able to survive. And so they're looking for something, anything out there that might help them survive. The cell that takes up the soluble DNA has to be made competent so that it's capable of accepting the genetic material through the cell wall and cell membrane. And so here's a cell that dies. And when it dies, the DNA is usually chopped up into little pieces, making it soluble in the solution. This other cell is undergoing some sort of stress, and it's going, what's out there that might help me survive? And they go, oh, here's this little piece of DNA. I wonder what it does. So it brings it in, puts it in its chromosome, and then reads the genetic material, and perhaps that gene product will be something that will help this cell survive whatever the stressor is in the environment. And transformation was actually discovered in the 1920s in an elegant experiment by Frederick Griffith. He worked with two forms of Streptococcus pneumoniae, and we still have these two forms today. There's an encapsulated form, which remember I said the capsule was kind of like a cloaking device for the immune system, and non-encapsulated strep pneumoniae. The encapsulated strains tended to grow smooth colonies in appearance on media, where the strains lacking the capsule tended to have a rough strain. So they were known as S strains and R strains. I'll simply call them encapsulated or non-encapsulated. So what he did first was he grew a bunch of the encapsulated Streptococcus pneumoniae. He took a pure colony, following Koch's postulates, and inoculated susceptible hosts. And the mouse actually developed severe pneumonia and died. And he did re-isolate and said, yep, he got live encapsulated strains back out of the dead mouse. Then he took the non-encapsulated, grew them in a pure colony, injected into a mouse, and even though the mouse may have gotten sick, it didn't die and actually recovered from the disease, and nothing was actually recovered from the mouse. Then Griffith took some encapsulated strep pneumoniae in pure culture, and then heat killed it. He injected the heat-killed encapsulated strain into the mouse, and the mouse did not get sick and did not die, and he was unable to actually isolate anything from the mouse. But then he came up with this idea. He took live, non-encapsulated strep pneumoniae, took heat-killed encapsulated pneumoniae, and injected the combination into the mouse. The mouse got sick and died. And he re-isolated then both encapsulated living strep pneumoniae and non-encapsulated form. Well, what was going on in the mouse? The immune system could recognize the non-encapsulated strain and kill it. These were under stress of dying, and so what did they do? They looked around in the environment, seeing, is there anything out there that might give me some protection. And some of these non-encapsulated bacteria picked up a gene from the dead cells that were breaking apart, and some of those genes actually then were the capsular gene that allowed them to now grow the invisibility cloak of the capsule. Transduction is the case of piggybacking DNA. And really, transduction is going to use a bacteriophage. The bacteriophage is going to serve as the carrier of the bacterial DNA from a donor cell to a recipient bacterial cell. This occurs in a broad spectrum of bacteria, but the participating bacteria of transduction is a single event and have to be of the same species because the bacteriophage is bacterial cell specific. General transduction simply shows random fragmentation of disintegrating host DNA of the bacterial cell that are taken up accidentally by the bacteriophage during the virus assembly. Any gene from the bacterium then can be transmitted by the bacteriophage to another cell. So here a bacterium is infected by a bacteriophage. This forces the bacterial cell to start making viral proteins. And to do this, oftentimes, the bacteriophage will actually cause the chromosome of the bacteria to be chunked up in little pieces. And so as we're assembling these viruses in the lytic stage of the bacterium, most of the viruses will take up the viral genome, but some will just take up any old piece of DNA, and that may be a bacterial piece of DNA. So when this cell lyses, these cells, or these bacteriophages, all go out and infect new cells. Well, if it happens to be this one carrying bacterial DNA, it will infect its host cell, but not cause viral replication, but that now new or that piece of bacterial DNA may be incorporated into the now recipient cell's own genome and change the genetics and cause a recombination within this cell. Specialized transduction is a more highly specific part of the host genome, is regularly incorporated into the virus genome. Really specialized transduction, as the virus is being assembled, we get a little piece of bacterial DNA and some of the viral DNA as well. This is explained by the existence of a temperate prophage that's inserted in a fixed site on the bacterial chromosome. In other words, what we can see is even in normal bacterial cells, a small amount of the bacterial chromosome is actually viral in origin. So the prophage DNA separates from the chromosome and can carry the host genes with it. So what we'll see is as again these viruses are being assembled, we actually will cut the bacterial chromosome and we get a little bit of viral genome and bacterial genome together. When that combination that is injected into a newly infected cell, it can be incorporated completely into the bacterial's genome, or only the prophage may be incorporated into the gene or into the chromosome. Now, one other thing that we can see in bacterial cells is something called transposons or transposable elements. These oftentimes are referred to as jumping genes because they can shift easily from one part of the genome to another within the same cell or to another cell. And it was first proposed by Barbara McClintock in 1951. And these jumping genes are actually widespread among cells and even viruses. Our own cells even have transposons or transposable elements. So here's a transposon, and it could simply move from this area over to this area. Or we could actually move it and copy it so that we move copies of it around. Or we could move it to the plasmid, and the plasmid then could be transferred to another cell, either through conjugation or another method. Insertion elements, these are the smallest transposons that exist, and they consist of two tandem repeats within the genetic code. And I'll show you a picture of that in a moment. Retrotransposons are a type of transposon that can transcribe DNA into RNA and then back into DNA for insertion into a new location. Other transposons contain genes that code for antibiotic or toxin production or even resistance to something. So here's an example. The transposon gene will be found between something known as inverted repeats. So in other words, if you read the 5' to 3' direction, GGCCA, etc., and you go to the opposite side, reading from 5' to 3', you should see GGCCA. They are simply inverted repeats, or what we call palindromes of each other. In the host DNA that we're going to put this transpose into, we have, again, areas that are going to be inverted repeats. And so what we'll see is that we have enzymes that will cut the DNA. And what will happen is then the transposon will fit into the cut ends of the DNA. We fill in the gaps here, and now I have moved or inserted this transposon into a different area. And we wind up then with direct repeats. And again, so we have ATGCA. If we go to the other end, the same thing, ATGCA. They are being read in the same direction on the same side. General effects of transposons: they will scramble the genetic language. They can be beneficial or adverse depending upon where the insertion occurs within the chromosome, what kind of genes are being relocated, what type of cell is involved. Effects of transposons in bacteria is sometimes we'll see changes in the colony morphology or pigmentation of the colony, or even antigenic characteristics of the bacteria. They can be used to replace damaged DNA. It can also be involved in intermicrobial transfer of drug resistance, especially if the transposon that's carrying an antibiotic resistance gene is inserted into the plasmid. The cell undergoes conjugation. Now the recipient cell has that gene for that antibiotic resistance. Pathogenicity islands have the ability to make their hosts more pathogenic. Typically, these pathogenicity islands contain multiple genes that are coordinated to create a new trait in the recipient bacterium. These islands are flanked by sequences that look very much like the transposon genes and will be cut. So our next concept check here. So first of all, what are the 3 ways that bacteria can horizontally share genes? What's the difference between the F-factor transfer and high-frequency transfer? A cell must first be what in order to be transformed? And what are involved in transduction? And what's the formal name for the jumping genes? The next thing we need to discuss are mutations, or changes or errors within the genetic code. So a mutation is any change or error to the nucleotide sequence within the genome. And really, mutations are the driving force of all evolution. In microorganisms, mutations become evident in altered gene expression, such as, again, a pigment change of the colony, or development of the resistance of a drug, or some other gene change. Wild type of bacteria or organisms exhibit a natural or non-mutated characteristic, and this will be the majority of that organism that's found in nature or in the wild. The trait is present in highest numbers within that population. A mutant strain is any organism that bears a mutation So what are some of the causes of mutation? Well, mutations can just happen spontaneously. It's fairly rare because we have a number of proofreading enzymes, including the polymerases, but other proofreading enzymes as well. So when we match an A with a T or a U, that's usually going to stay that way. However, sometimes cytosine tries to match with A, and so could we get a spontaneous mutation? It can happen, but it's fairly unique in nature. Induced mutations, however, result from exposure to known mutagens, and mutagens are anything that will cause a mutation. A carcinogen, for example, is a mutagen because carcinogens cause a mutation within the cancer cell that will cause cancer, but not all mutagens are carcinogenic. So here's just a table with some of these, and you can read down through these, but there's a whole number of chemical mutagens out there, and of course radiation, including both ionizing and non-ionizing radiation. Now, we can categorize mutations into two primary groups: point mutations and frame shift mutations. Point mutation is a small mutation that affects only a single base within the genome. It involves addition, deletion, or substitution of a single base. Lethal mutations can actually lead to cell dysfunction and death, and frame shift mutations oftentimes are lethal. Neutral mutations are usually point mutations that produce neither adverse nor helpful changes. They're sometimes also known as silent mutation. So let's take a look at a missense mutation is going to be anything that causes another amino acid to be put in an area where something else was supposed to be. So we actually wind up with a change in the amino acid sequence. This is a type of point mutation. A nonsense mutation is where this changes a normal codon into a stop codon, thereby prematurely stopping protein synthesis. This can be due to either a point mutation or more oftentimes it's due to a frame shift mutation. Silent or neutral mutations alter a base but does not change the amino acid sequence, therefore has no effect in the protein. And this is due to, again, that redundancy and wobble within the genetic code. Usually these are point mutations. A back mutation is kind of interesting in that this can occur in a gene that has undergone a mutation, but then is reversed to actually go back to the original genetic code. Frame shift mutations occur when one or more bases are inserted or deleted from a newly synthesized DNA strand, and this shifts the reading frame of the messenger RNA. So from the point of mutation on, we now have new codons. This nearly always results in a non-functional protein. Every amino acid after the point of mutation is different from what was coded for in the original DNA. Insertion of bases in multiples of 3, however, does not disturb the reading frame. It simply puts in another amino acid within the sequence. So here's our normal gene. Here's our DNA template. There's the messenger RNA. AUG codes for methionine, ACC, threonine, etc., etc., etc. So this is our normal protein amino acid sequence. In this one, however, instead of CTG in the DNA, where we get GAC coding for asparagine, we'll change that CG pair original in the DNA to an AT pair, and we now wind up with GAA that codes for glutamamine instead of asparagine. This is a missense mutation. But you will notice, however, every amino acid after that actually remains the same. So this may cause a problem in the protein structure and function, or it may not. It kind of depends upon one, where the mutational amino acid is located, and does it change the folding of the protein that much. This mutation, instead of having CTG in the DNA, we now have CTA. This causes a messenger RNA codon of GAU, but that codes for asparagine as well as CAG. So this would be a silent mutation. There's no change in the function of the protein. Now, again, frame shift mutations. These shift the reading frame. So let's compare our DNA here to the original. TAC, TAC, TGG, TGG, CTG. Oh wait, what we got was we put a guanine in here. And what that does now is shift everything. So the C is here, now it's in the middle. The T was in the middle, now it's on the end. And this G now starts the next triplet or codon. And so what we'll see is that now we affect every amino acid after the point of mutation. And this is going to then make an entirely different protein that either is not functional at all or functions very poorly. This one actually shows a frame shift mutation that results in a stop codon being produced. So we come along and we replace the asparagine with threonine, the glutamic acid with arginine, but now we have ACT, and this is the ACT there from the frame shift, and that codes for UGA, which means stop. And this is going to cause the failure of the protein to actually be made. Now, again, sometimes these nonsense mutations can actually be caused by a point mutation as well. Can we repair mutations? Well, we actually can. Oftentimes, if we have damage from ultraviolet light to the DNA, we'll have a group of enzymes that do photoreactivation, and this actually simply repairs the damage caused by that ultraviolet radiation, and we'll talk about that a little bit later on as well. Excision repair, this is mutations that are excised by a series of enzymes that remove the incorrect bases and pair in the correct ones. So here's our one side of our strand of nucleic acid, and you notice that these bases don't fit. So these excisional enzymes will come in and simply cut out those not correct nucleotides, and we'll have an enzyme then that comes in and puts in the correct nucleotides. The problem is, if we're making DNA, which side was the original side? So if we come along to a scenario like this, was this the original parental strand and therefore the quote correct strand? Then we make the correct repair. But what if this one was the correct parental strand and these were the errors that were made? Well, now we're actually cutting the parental strand and putting in the bases that match the mutation. And this can then result in mutations within the genetic code. So we have a 50-50 shot of correcting it back to the original strand of DNA. Mismatch repair. This is a repair system that's usually primarily found in those polymerases that can locate the mismatched bases that were missed during the proofreading process. The bases do need to usually be replaced soon after the mismatch is made, or oftentimes the repair enzymes won't be able to recognize it and it will remain. The Ames test, I want to mention it here because this is a commonly used test to rapidly detect chemicals with carcinogenic and therefore mutagenic potential. This uses bacteria rather than experimental animals in testing for the carcinogenic potential. It allows easy observation and monitoring of the gene expression and mutation rate of the cancer cells. Any chemical capable of mutating bacterial DNA could similarly mutate mammalian DNA and therefore is potentially hazardous. So this is kind of a quick and dirty test. We use actually a specific type of mutant salmonella, and this is a salmonella that is histidine dependent, meaning that this particular salmonella cannot make its own histidine like the wild type of salmonella can. So histidine has to be added into the environment in an enriched media in order for the histidine negative salmonella to grow. In the control setup, bacteria are plated on a histidine-free medium containing liver enzymes but lacking the test agent. Because there is no histidine in this plate, we would expect none or very few colonies to grow. Sometimes we will find in these mutant strains that they back mutate to the wild type and therefore can produce their own histidine and grow. So we may have a few colonies that back mutate. But if we put the liver extract and the histidine negative salmonella and the potential carcinogen in this and plate it with no histidine, and we get lots and lots and lots of colonies growing, that means something mutated the histidine negative strain to a histidine positive strain. And so we will say that whatever the chemical we added is what caused the mutation from histidine negative to histidine positive. Now, the Ames test is not perfect, I will mention that, and so if we do find something that is found to be potentially carcinogenic on the Ames test, we will further study it in animal models. Now, many mutations will not be repaired. The effects of mutation depends upon the nature of the mutation and the strategies available to the cell or organism for dealing with the mutation. Mutations, though, that are not repaired will become permanent and heritable to the next generation. These will be passed on to the offspring of the organisms and new viruses and become a long-term part then of the gene pool of that organism. A small number of mutations contribute to the overall success of the individual and the population. And again, this is the main driving force behind evolution. Variant strains with alternate ways of expressing a trait can more readily adapt, survive, and reproduce, where older non-muted strains may actually die off in the environment they find themselves in. So, last concept check. True or false, a spontaneous mutation arises from exposure to chemical or physical agents. What type of mutation results in the stop codon? What type of mutation nearly always results in a non-functional protein? And the what is a first-line screening technique to detect mutagenic chemicals. And true or false, mutations are always harmful to organisms. And that's it for this chapter, so see you in the next one.